Hi, my name is Chloe Blunk, and this is the week 3 lab video for microbiology. It's just reviewing how to use a microscope and just getting comfortable with it since we're going to be using it for the next few weeks. Today is July 19th. It's early in the morning right now, so good morning everybody. So I just quickly wanted to review the parts of the microscope. Up here we have the ocular lenses. This is the arm of the microscope. It's just there to hold, have a strong base line on it. We have the turret right here, which is what we use to rotate to change our field of vision and the magnification. We have two of the fine knobs right here. The bottom one is for like the fine micro adjusting. I didn't really use it that much. I'm not a big fan of it, to be honest. We have our stage, and then we just have the adjusters right here. And then the green one, these are color-coded. This one right here is the scanning lens, and then it goes from scanning to low power, which is the blue one, and then to high power, which is the red one right here. And then I put it away, but then we have the oil immersion lens, which is just a special lens that you can use to help with the light refraction. You can't use the oil on the other ones because it can cause a lot of damage, and I noticed that it was not my favorite to use the oil immersion lens. It did make a difference viewing the slides with it, but it was just a pain in the butt as far as having to clean it up after every slide. So it wasn't my favorite, but it's definitely very useful. And then we just have on the bottom right here, and this is where our light's going to be. And then we just place it underneath. And then it kind of acts like our corneas right here. Where we can condense the light. It's the condenser, so we can open it up if we want full light, and then we can close it up if we want very minimum light. Didn't really have to use that much, except for one slide that was not stained where we needed to use it. But otherwise, it's pretty self-explanatory. It kind of took a bit of adjusting and just using techniques of how to put the slide in onto the stage and utilize the adjustment knobs right here to move everything side to side, up and down. So that kind of got some taking used to. So that was the first exercise, was just going over parts of the microscope and learning how to use it by taking the measuring tape. And the second part was reviewing the slides that came in a box of different bacteria, and then we also had to use the letter E. Again, the oil immersion was not my favorite. There's just a lot of cleanup after each slide, and just getting used to working the microscope. But it was fun to watch the bacteria and just to see how much farther in depth you can see the tiny little, it just turns into like bubbles. I feel like at first it's a big picture, and then you just see little bubbles everywhere, and it makes a bigger picture. And then on the third exercise, it was the cheek smear and the dental smear. It wasn't that hard. It was pretty quick and easy to go through. They're pretty similar. From what I remember, one of the questions was the similarities and differences. Similar in the fact that they were not stained, so you're literally looking at just black and white and then water in the background. So I had a hard time I could still see the water kind of shaking when I would change the stage to move the slide around. And then the only differences was with the cheek smear, it was very small clusters of cells, whereas on the dental smear with the tartar, it kind of looked like little spikes, kind of like little thorns that you see in the desert that you step on. That's what it reminded me of. But that is it for this lab for going over how to use a microscope.